to the rest of LNS, and I heard the word biogeochemistry float in the air. That always perks my ear as somebody who is a biogeochemist once in a while. And I said, oh, who are you? And we started chatting, and it occurred to me that what the work that Athena does fits very squarely in the work that we like to showcase in the Climate, People, Environment Program Seminar, and the types of work that we do in the Center for Climatic Research. And since you're across the street, it seemed like an appropriate time to give us a chance to learn a little bit more about your work and maybe find some connections. And I did get a formal bio for you, so I should read it. Dr. Nghiem is an assistant professor. He started this year at UW-Madison in Geoscience, and her research focuses on the cycling of trace elements and contaminants across different reservoirs in the environment, including arsenic, which has impacts on human health. And then before she arrived here in the fall, she held an ETH fellowship at ETH Zurich with a guest appointment at EAWAT, not sure how that's pronounced, the Swiss Federal Institute of Aquatic Science and Technology. And she did her PhD at Columbia University and the Lamont Doherty Earth Observatory, where she was an NSF Graduate Research Fellow. And so thank you for making the trek across our weird patio that someday will turn into a building. Turn it over to you. I hope to see more of you.
Thank you so much for the introduction, and I'm very excited to cross the patio today and finally be in this building to present to you some of my work focused on tracing the dynamic distribution of arsenic across reservoirs. So as many of us know and love the water cycle, of course, water moves through many different reservoirs in the environment, and it's not constrained to only one reservoir. Similarly, we think about elemental cycles like carbon, which also move through different parts of Earth's environment and are not constrained only to a certain reservoir. Something that I think about in much smaller magnitudes is the arsenic cycle, which also similarly cycles throughout the environment with different sources and fluxes. And why is thinking about arsenic so important, even though it's so much smaller in magnitude than the carbon cycle? Well, the motivation is that not only is arsenic a trace element that is present naturally in the environment that undergoes this biogeochemical cycling, but moreover, arsenic carries the most significant potential threat to human health because of its frequency, toxicity, and potential for human exposure. So this kind of exposure can happen in different ways in the environment. For example, pathways for exposure that lead to adverse health effects, some of which can include multiple types of cancers or cardiovascular disease, and others include perhaps the well-known consumption of naturally occurring or geogenic arsenic-contaminated groundwater. So not only is groundwater used as a drinking water source, which then affects and exposes millions of people to arsenic contamination, but it can also be used for irrigation onto surface environments. So then arsenic can get into the soils as well as plants, which leads to another exposure source, the consumption of arsenic-containing food. So many of you might have heard about arsenic in rice, for example. And then maybe a less thought about exposure source of arsenic into the environment is the inhalation of atmospheric arsenic. So actually this surface environment is very dynamic. Once arsenic enters the system, microbial communities and plants can also volatilize arsenic into the atmosphere, as well as you can have deposition from other anthropogenic sources of arsenic. So there's many different exposure routes, some of which are localized, like selecting your drinking water or the food that you eat, and others are more sources of global exposure. For example, you can't really select the air that you breathe. So this gives us a picture of the different sources and sinks of arsenic in the environment. And in particular, something that I want to emphasize is that sometimes we have natural sources of arsenic, for example, arsenic in groundwater. It's naturally occurring, but actually our exposure is really modified by this anthropogenic flux. So the fact that we use the groundwater as a water resource, and we pump the groundwater out of its kind of storage in the groundwater environment leads to its exposure. So really the first step to understanding exposure is understanding the distribution of arsenic in the environment. So today I'll present to you two kind of investigations of arsenic distribution. One will be really focused on localized sources of exposure, such as groundwater pumping, and kind of the biogeochemical dynamics at play that can lead to increased arsenic in your groundwater. And maybe zooming out to something really different but maybe relevant for people here in this seminar is also a side study that we did started at ETH, which was more looking at the dynamic reservoir in the atmosphere of arsenic. So let's first zoom in on this small scale groundwater pumping. For those of you who are not thinking about arsenic all the time like I am, just a brief background review. Arsenic released to groundwater is commonly attributed to the microbial reductive dissolution of arsenic-bearing iron minerals. So when you have sediments that are deposited in the anoxic subsurface, you have in this very simplified schematic, you have your iron-reducing bacteria, and then for their metabolism, they couple the reduction of these arsenic-bearing iron-3 minerals to the oxidation of organic carbon. So in this process, the iron-3 mineral is reduced to dissolved iron-2 and thereby releases arsenic into solution or into your groundwater. Of course, as I said, this is very simplified, so there's a lot of multiple factors that can influence what actually happens in the environment, including but not limited to hydrology and mineralogy, and I can make this figure more and more complicated as we go, like aquifer composition, the different types of mineral, the subsurface stratigraphy, so more mineral formation. So this really creates large variability at all scales. And we can kind of start to see this variability when we look at groundwater arsenic concentrations across South and Southeast Asia, which is a really hot spot of arsenic contamination. This comes from a USGS Powell Center working group that we tried to compile all the past studies on arsenic in groundwater from field sites and different papers to geological surveys that occurred. So anything that's not in this darker blue color, anything above 10 is above the World Health Organization guidelines for arsenic in drinking water. And what you start to see is that some countries have a lot of points, and you're like, oh, maybe it's not so bad. It looks mostly blue. It should be safe. But one really characteristic thing about contamination of groundwater, especially with arsenic, is this kind of small scale, local scale heterogeneity. So when you zoom in on a scale that's really relevant for water usage, like at your household scale, you can see that your well might be blue, might be good, and low in arsenic, but your neighbor's well might be really high in arsenic. So this shows you the scale of heterogeneity that occurs when you look at groundwater arsenic. Of course, right now I'm only showing you the 2D view, so all these wells have different depths, of course, and this is also another important thing to consider. So when we now we're looking at the country of Bangladesh, we see that spatial variability also exists with depth in this movie. So you can see that there's still variability of the arsenic concentrations, but we start to see kind of this general pattern or trend that emerges. And what we see is that, at least for Bangladesh, we can say that there's a broad trend in arsenic concentration with depth. So we see we have a lot of low arsenic or blue points down here. And similarly, I'm just showing you the same thing, but kind of in one well where we have really high arsenic concentrations in the shallow subsurface, and then going deeper with depth, the arsenic concentrations are really low. And this is not just a coincidence, but actually the difference in arsenic concentrations is associated with sediment age. So where we have this higher groundwater arsenic, it's associated with younger or these Holocene age aquifer sediments. Then we have these lower groundwater arsenic that is associated with these Pleistocene age aquifer sediments. And this is not necessarily because of the age of the sediments itself, but rather what it's associated with. So these Pleistocene aquifers contain more oxidized sediments, while Holocene aquifers typically contain more reduced sediments. So in terms of iron mineralogy, for example, when you drill a well, you might find these orange sands. If you drill into these Pleistocene aquifers, they're typically indicative of your iron-3 minerals. Meanwhile, if you drill a well into a Holocene aquifer, you might typically find these gray sands, which are representative of iron-2 minerals. So this is important because of the redox state of the sediment and thus its association with groundwater arsenic. So when you have these oxidized iron 3 minerals, arsenic can sorb onto these minerals, and thus you have less arsenic in groundwater. Meanwhile, when you have these iron 2 minerals, that means the arsenic has been already released from these minerals, or the arsenic cannot sorb as well onto these iron 2 minerals any longer. So this kind of leads to the conclusion that Pleistocene aquifers are assumed to be safeguarded against this arsenic contamination due to these abundant oxidized iron minerals. And then people commonly rely on these deeper Pleistocene aquifers that are believed to be less contaminated as a cost-effective method to reduce exposure to high arsenic. So for example, if your well is high in arsenic in Bangladesh, they would have a little placard on it, and then you would switch to a different source of hopefully low arsenic well near you. So you would think that now this is solved and we're done with thinking about groundwater arsenic. However, something that starts to happen where groundwater with low arsenic is available, the sustainability of these uncontaminated aquifers are currently being jeopardized, for example, by groundwater pumping. That can cause the infiltration of high arsenic into these low arsenic aquifers. So one scenario where we really see this is also in this super simplified schematic. This is a scenario that is a potential threat across Bangladesh, where you have your higher groundwater arsenic, but large-scale pumping by, for example, the city of Dhaka with its huge population growth and its need for water resources from groundwater has created a cone of depression. And this can lead to the infiltration of this high arsenic groundwater into these Pleistocene aquifers that are supposedly safe. So in this first investigation of arsenic distribution, the goal was to determine what mechanisms and biogeochemical processes occur when this high arsenic groundwater infiltrates into these low arsenic or safe aquifers. And specifically, how does this kind of assumption that we had hold up about these orange aquifer sediments being safe? So here we're really looking at the small-scale biogeochemistry. We had an experiment that simulated Holocene groundwater infiltrating through these Pleistocene sediments as a result of anthropogenic exacerbation. There was kind of this tank contraption that pumped up groundwater directly in the field and kept it anoxic, and then we pumped it through sediment pores that were also drilled directly in the field. So this gave us the opportunity to do really a hybrid field column experiment, which is really nice because oftentimes when you go to a field site and you try to characterize it, there's a lot of unknowns and constraints that are hard to capture. Meanwhile, when you're in a lab environment and you want to create a scenario that represents the real environment outside, it's hard to get things like real groundwater. It's oxidized by the time you bring it back to the lab, or get sediments that are representative of what's actually happening with the, for example, microbial communities that are at play. So this was a really nice hybrid study that had the best of both worlds. We could monitor things in our experiment over time, but we could also capture the real-life heterogeneity that would be in the field. So this is just a diagram showing you that bag there. The groundwater was pumped up and put through peristaltic pumps. These were the cores that were actually taken in the field. And today I'll just focus on this, the Pleistocene or orange sands. So from this, we can measure a lot of things. And so in this column experiment, we wanted to look at the measurements and be able to describe processes responsible for arsenic release when this Holocene groundwater infiltrated these Pleistocene sediments. So we have our groundwater going in, our influent, some biogeochemical process happening in the middle, and then our effluent, which we can measure. And so some of the results look like this for the breakthrough curve. Essentially, I'll orient you to how this results look like. The x-axis is pore volume, which is just how often the column is flushed out. So just think of it as a measure of time. We could do 10 pore volumes per day. So this is around a 20-day experiment. And then on the y-axis, here we have arsenic concentrations. At the dotted line is the concentration coming in from the groundwater. And then the actual points that you see here are observations of arsenic that are coming out in the effluent. So this is like an arsenic breakthrough curve. So of course we didn't only measure arsenic, but also other parameters relevant to our cycling, including sulfate, iron, phosphate, because sometimes it competes with arsenic for sites on these iron minerals, and many other things that I don't show you here. So basically the approach we wanted to test was, can we test for different biogeochemical reaction networks with geochemical modeling that accounts for sorption, kinetics, and thermodynamics, coupled to the flow that is happening in the column. And we're taking this from measurements to models, so we also wanted to test this canonical explanation for arsenic release and see how well it fits our measurements. So just briefly to do that, of course we have our modeling of flow with ModFlow, and then I performed reactive transport modeling with PHT3D, which is combines the transport program, MT3DMS, and the geochemical model FreakC. And then we also did optimization and model sensitivity and certainty analysis with parameter estimation. And this allows for our conceptual models to be tested and optimized with our observations. And then we also performed model uncertainty analysis with linearized Bayesian methods. Yes. So what happens if we test our first scenario by including only iron-reducing bacteria? Well, when we put that in our model, the model with this iron-reducing bacteria only cannot explain our effluent concentration even after rigorous optimization. So you can see the model is the blue line, and arsenic looks pretty good actually if you kind of squint at it, and iron as well. Phosphate's not really doing anything, but sulfate, obviously we're missing an important process related to sulfate. So when we go back and think to our conceptual model, of course a huge decrease in sulfate usually implies sulfate reduction or sulfate reducing bacteria at play. So then we incorporate our sulfate reducers into our biogeochemical reaction network. This sulfate reduction, of course, when you have a sulfate reduction, you produce sulfide, and it just doesn't end there. So we have to complete the rest of the cycle. So we have some mineral formation from the sulfide, like iron sulfides and arsenic sulfides, as well as some other mineral transformations. And actually how we can kind of guess what processes occur before we do the modeling is that we measured the sediments pre-experiment with synchrotron-based methods such as iron extended X-ray absorption fine structure to find out about the iron mineralogy, and arsenic zane spectroscopy. So we kind of have an indicator of what happens, or what's pre-experiment and what's post-experiment, so that we can put it in our reaction network. And finally, to round out our other sulfur-related processes, we also added some other reactions, coupled reactions that could potentially occur. So altogether, we have our biogeochemical reaction network, and reactive transport modeling then allows us to test this part of the previous model that we had with incorporation of everything into our biogeochemical reaction network. So when we did that, what we found is that the full biogeochemical reaction network is needed to explain the effluent concentrations. Here we get a much better fit for arsenic, but we also, importantly, can fit this decrease in sulfate and keep the other parameters still fitted as well. So we need the full biogeochemical reaction network, but now that we have this full model, another thing we can do is then test different parts of the model and see how important they are overall to our fitting to our observations. So something I kind of glanced over or skipped over before was the inclusion versus exclusion of these thioarsenic species, which are arsenic with an attached sulfide group or sulfur group. So essentially when we think about arsenic in the environment, sometimes we usually think of it as arsenic oxyanion, so only bonded to oxygen. However, once you replace one of the oxygens with at least one sulfur, they're called these thioarsenic species that are also aqueous. So what happens if we turn this part, or thinking about arsenic speciation, on and off? Well, without the inclusion of these thioarsenic species, for example, the no thioarsenic model, arsenic breakthrough curve cannot be explained. So if we see here, there is slower breakthrough of arsenic, and it doesn't match the arsenic breakthrough as if we included the thioarsenic species. And actually, the reason for this is because when we have our typical arsenic oxyanions here, as I mentioned, they're bonded to oxygen. when we have our thioarsenic species, they actually have low or no sorption onto iron minerals compared to arsenic oxyanions. So here you can see the arsenic is still sorbed onto my little cartoon of the iron minerals, but these thioarsenic species have faster mobility because they're not as easily sorbed onto these iron minerals. Thus, they're more mobile than regularly considered species of arsenic oxyanions and then lead to faster contamination, showing you this faster breakthrough of arsenic. So in the end, the main result of this work is that when we had this limited sulfate reduction, it led to all these extra sulfur-related processes, and it's sufficient for rapid contamination of groundwater with arsenic, even in aquifers with oxidized iron minerals, that we would assume to sorb these arsenic species onto their surface. And ultimately, this can lead to faster redistribution of contamination of groundwater, which increases public health risk, especially as groundwater pumping increases throughout the region. So kind of the outlook for this work is that was a very small scale study in a column experiment. So we are now considering the generalizability of these biogeochemical reaction networks and how to accurately characterize arsenic mobility and contamination. And some of this work is part of an ongoing project that I have with collaborators in Switzerland, and we're working on a project in Vietnam trying to look at decadal scale changes and thinking about maybe the importance of considering arsenic speciation in relating these decadal scale changes to groundwater pumping in the Red River and the Mekong Delta. So that is a little past work and a preview of future work. But now I'm going to jump like a very, very large to a very large scale from this local redistribution to global redistribution. So now we're going to jump to the atmospheric reservoir, maybe something a little different to think about arsenic cycling in the environment there. So as many of you probably already know, the atmosphere is a globally relevant and also a dynamic reservoir. Arsenic can also undergo long-range transport from multiple natural and anthropogenic emission sources, and it was previously estimated that there was a lifetime of 4.5 days, and it could be redistributed from local sources to the global environment much more faster than in groundwater. So some of the previous emission sources of arsenic include coal combustion, smelting and metals production, mining, burning for agriculture and waste incineration, burning of fuel and wood sources. Also, arsenic was used as a pesticide for agriculture, and gas from landfills. So there, however, also exist natural sources of arsenic into the environment. So volcanoes are a known source, dust and wind erosion, forest fires, and sea spray. But also biogenic sources, for example, due to biological volatilization from long postulated terrestrial sources and more recently emphasized marine sources. So together you can see the different natural and anthropogenic sources in the environment, and fires are kind of a little bit of both. So when we looked at the past literature for estimates of these emissions, past estimates for anthropogenic and natural emission fluxes have really large ranges. So here, if we start in the box models a long time ago, we can see a lot of variability. Of course, a lot of times these box models were based on a few measurements, and so they were the best estimates at the time. And now there's been some more recent atmospheric models that I'll talk about more later. But in general, you can kind of see this large variability in both the natural and the anthropogenic emissions. However, since a lot of these initial box models were estimated, we've made a lot of progress in research. Since these initial estimates, both experimental and computational methods have greatly advanced. We have better measurements of trace elements, for example, with different inductively coupled plasma mass spectrometry systems, as well as the ability to do way more complex atmospheric model simulations. So if we start on the side of looking at the better measurements of trace elements, we can see what we now know, thanks to the great work in measurements. Not only are there better measurements of totals in trace elements, for example, lower detection limits when we have your ICP-MS methods, there's also been a lot of progress in speciation, as we just saw talking about speciation in the previous little story. So again, here are your arsenic oxyanions. And when we think about the atmospherically relevant species, we have some other species that I'll introduce to you here. We have our volatile species. So these are our arsine and our methylated arsine gases. So arsine is a very toxic gas. Some of you might know already. But the methylated arsines also come from biogenic sources, and it's been postulated that trimethyl arsine is kind of the final biogenic product. After you have emission of these from different emission sources, they undergo oxidation. So they're oxidized into their low volatile species. You can see the complicated acronyms here. And then since they are of low volatility, they undergo condensation and then deposition onto the surface environment. So some previous work by the group that I was in at ETH showed that trace element speciation is influenced by long and short range atmospheric transport. So in the work of Esther here, she demonstrated that she found 5 to 85% methylated species in a 5-year aerosol species. She also sampled rainwater, cloud water, and did some other speciation. But essentially this means that there could be some biogenic sources existing out there in the environment, and she took these measurements at a high altitude observatory in the mountains in France, which looks really nice right here. Okay, so that's on the measurement side. Now going to models. Of course now with our high performance computing powers, we have the ability to do more complex atmospheric model simulations. So atmospheric models, as you know, can simulate atmospheric chemistry, climate, and transport. And using our proposed and known emission sources, these atmospheric models are validated against the measurements such as the ones I showed you from Esther, like aerosols and depositions. So actually there was a model of arsenic in the atmosphere before, but actually they only emitted arsenic as PM 2.5 and then also transported arsenic as PM 2.5. So none of this volatile kind of schematic that I showed you before. And this is important because models are only as good as incorporation of measurements. So one thing that they did not yet consider in their model at least was the natural biogenic emissions. So actually natural biogenic emissions of arsenic by organisms have been known for over 100 years. So back in the old days, they actually used arsenic as kind of this green coloring, and it was found in wallpapers, and what they didn't know at that time was microbial community and fungi could take up this arsenic and release arsine gases, poisonous gases, into your room. So it was already known back then that arsenic was poisonous and could also emit these gases from wallpapers, paintings, and fabrics. And now more recent measurements we have shown in Esther's work and the other groups have shown that biovolatilization occurs from these biogenic sources in the environment, either by measuring the emissions themselves or by measuring the deposition. So this is important because these recent atmospheric models miss these important biogenic emissions and indeed actually only included anthropogenic and volcanic emissions. So when we kind of break down the variability between these different emission sources, we can see that maybe the variability in anthropogenic emissions over time might be actually due to the temporal changes that we could expect in anthropogenic emissions. But the spread in natural emissions likely reflects their uncertainties in their estimate or potential legacy impacts. So there's large variability in these biogenic emissions and their contributions to natural emissions. So here's some estimates together from literature. You can see this huge range for terrestrial biogenic emissions and an even larger range for the marine biogenic emissions. So why also do we care? Actually, as I mentioned, these anthropogenic emissions are indeed changing. And here we can see the emissions of sulfur or sulfur dioxide and related species and arsenic relative to the year 1990. And you can see that, at least in Europe, there's a decrease over time. And as anthropogenic emissions decrease, the importance of biogenic emissions may increase. Another important thing about anthropogenic emissions is that it might be just the entry into Earth's surface system, but arsenic can continue to be recycled, especially via biological processes. So we wanted to understand what's the impact. Some of you might have heard of similar things happening for mercury, and they call it legacy mercury, so I have a question mark here. Maybe there's legacy arsenic in the environment. And the emission trends are always not straightforward as well. So here we have the similar figure, but now we added some more countries onto there. And they're not always also similar to the deposition flux. So again, we have this decrease for Europe and now North America, which also matches the decrease in arsenic deposition flux. However, if we look at Asia, we can see that China has started to decrease their emissions, but India has this huge spike. So this probably leads to the discrepancy in these deposition fluxes in South and Southeast Asia. So maybe in this case over here, there could be long-range transport from other regions, such as from coal burning in India, seasonally varying biomass emission or burning in South and Southeast Asia, or just the re-emission of anthropogenic arsenic via these biogenic processes. So for our approach to investigate this further, we use this model ICAN-ART. ICAN stands for the Icosahedral Non-Hydrostatic Model, which is a global weather and climate model with flexible framework allowing for regional grid requirements because it's based on this triangular grid cells, and it's developed by the German Weather Service and the Max Planck Institute for Meteorology. And ART is the extension that accounts for aerosols and reactive trace gases developed by colleagues at Karlsruhe Institute of Technology. And so with this framework, at UTH we try to incorporate arsenic. So as I mentioned, there was anthropogenic and volcanic emissions in previous models, but no speciation, atmospheric reactions, or chemistry. So we wanted to incorporate those into our ICAN-ART modeling framework. So first, we looked more closely at speciation, and these are the species that we ended up including, as I showed you before, the volatile species. And then we also wanted to simulate what happens after they're emitted with the chemistry. The hard part, as I mentioned, is that arsine gas is toxic, so there's not a lot of experiments in the literature with this gas. It's also very dangerous, so there's no rate constants, there's no reaction rates. It's very hard to put into the model. Luckily, one thing that did exist in the literature was kind of these stability constants of these different volatile species. So how we implemented it into the model was just to model, after emission, these species with first order decay with a rate constant, and then assuming after a certain time, after they're fully decayed, they would be converted into their oxidized forms. So here's just an example of the study that we used as basis. At least it was done under dark and light conditions, so we can simulate those in the atmosphere, and then you can see that the difference between the decay of each of these species and how fast they decay or can be transported in the atmosphere. So after the decay, you have your volatile species. They're oxidized to the low volatile species, and we wanted to test the simulation. So here we just have a very classic point source example to test arsenic chemistry. Volcanoes, as I mentioned, are a source of arsenic, and typically what was measured before is that it's a source of arsine gas. So we had this point source simulation that shows emission of arsine into the atmosphere, and then kind of its conversion partially to arsenate over time, just as a quick test. Okay, so now we said that now we can have the emissions and the conversion to oxidized species, so we can account for the chemistry somehow. The next thing that we accounted for from this diagram was then taking these species, and since they're low volatility, simulating their condensation to aerosols, and then eventually their deposition. So for this, we also did another volcanic simulation, so it'd be easy to see everything that changed in the model results. Here's an actual volcano that erupted in 2019. They didn't measure arsenic here, but we just simulated it. They were able to measure and model sulfate aerosol emissions, so this is also what we based it off of. So here we have our arsine gas in one time point in the emission, and then we had a first order decay to our arsenate, and then we had our condensation onto aerosols. So you can see some of the arsenic start to attach to aerosols that are in this simulation. So that was the check mark for condensation before deposition. And now my colleagues at ETH are finishing up this emissions scheme, especially because even though I talked about uncertainty in chemistry, previous models of atmospheric trace element cycling for another trace element, selenium, found that most of the uncertainty was actually mainly attributed to emissions. So even though we don't have all the reaction rates in chemistry, it's overall more important that we get the emission sources correct. And then some advantages with this model is that we can zoom into specific regions with the same model. So instead of needing to build a different model and a different code for the regional scale, we can automatically just zoom in and do a finer grid spacing for certain regions where we're most interested in. And this is where it will link back to kind of what I was talking about before with this surface environment. And one of the critical places that we'll actually think about this distribution is in rice paddy environments, because arsenic is an important threat and a dynamic trace element there. Not only from thinking about groundwater, but if we now transport back to the groundwater, we can see that the high arsenic in groundwater is in the same regions as irrigated rice paddies. So on the left-hand side, we have rice paddies in irrigated areas, and we have populations here living with likely high groundwater arsenic. So groundwater arsenic hotspots really coincide with where you have rice paddies and where they're irrigated by potentially this groundwater contaminated with arsenic. So this leads to another super simplified schematic, but really connecting all the parts of the cycle together, which is what I'm really interested in investigating further. So of course here we have our groundwater contaminated with arsenic, this very rudimentary pump. We use it to irrigate these surface environments and surface soils. A lot of people have investigated kind of the plant uptake, which is not my specialty, so I won't talk so much about it, but of course arsenic can be used by microbial communities here, or it can be in the plant roots and uptaken by the plants, can be volatilized in the environment, but also there could be deposition of arsenic from sources elsewhere. So kind of my interest area are really in characterizing this surface mass balance here with what we know about groundwater arsenic, and then hopefully the modeling results from that will be finalized by our colleagues, or my colleagues. So here now I'm just showing you again from that model, which actually only considered anthropogenic emissions. At least they show that there's a lot of deposition similarly also in the same regions that we also have these irrigated rice paddies. So that look is to stay tuned for the results of that work and see what results come out next from that. And with that, I just have my acknowledgments of the work, including my funding source, my ETH fellowship, and the time that I spent in the group at ETH and EAVAG. And with that, I'd be happy to take any questions.
Thanks. Definitely have some time for questions, so open it up to the floor. If there are students in the audience, we always prefer the students first, but maybe we're not. All right, we'll open it up to everyone. I'm just going to start. I'm fascinated by this transport idea of some of these biogenic aerosols you mentioned, and some of the low volatility ones that would be condensates. Tell me about cloud transport. Like, is there much, do you think, or is it pretty faster lifetime that we're talking about here?
Yeah, that's a good question. I think it's been so not considered yet for cloud transport. I mean, these species are measurable in aerosols and wet deposition, which kind of gives us the idea that they would be important. I know that Esther also measured cloud water in her, she also measured cloud water, but now I don't remember the proportion of it that was in cloud water.
Yeah.
But I could definitely send you her paper.
Yeah, it's just be curious, because I think it complicates the modeling.
Yeah,
too,
exactly.
Right? Because then you need to get that right into the transport as well.
Yeah, I think what we're aiming to do after the emissions are implemented is compare all the possible measurements that are existing of arsenic in deposition. So we, like for selenium, which atmospheric modeling has been done on this trace element, they found that most of the selenium was in wet deposition. And so they tried really to compare against wet deposition networks, but of course aerosol networks are more abundant than the measurements of wet deposition, especially for trace elements. So once the model is finalized, there will be comparison against both the aerosol and wet deposition measurements, and then we'll check the uncertainties in different aspects of the model parameters and try to calibrate the model and separate the measurements into both a test and training sets that as not to over calibrate. Yeah.
I was just curious how appropriate the use of the PM 2.5 tracer to transport these species is. Is that, likely to underestimate certain kinds of things or overestimate certain kinds of things?
Yeah, that's a great question. Now I can't find the slide. Yeah, that's a great question. I think there, in the previous modeling results, they did it with Geoscience, and what they found was they could match mostly. They couldn't really match the measurements of the aerosols in the end in the PM 2.5. I think the reason why they picked to emit it and transport it as PM 2.5 is because of the lack of information on arsenic chemistry, and also the fact that, the lack of information in chemistry, and also the fact that they were only looking at totals and they didn't have any information. They didn't have as much information on speciation, and speciation is the indicator that there could be different sources. And previously, I think they referenced another paper in the literature that said, "Oh, arsenic is mostly anthropogenic" in one of these kind of variable box model studies, and then they used that forward as their reasoning to not include other sources. So I think there's just been some historical things in the literature that have propagated and led to their implementation of arsenic as PM 2.5.
So I'll ask a question, even though this is far from what I understand is, so there's a lot of time spent on these transport models, right? But is it really predominantly localized when you're thinking about the groundwater and the rice paddy fields, right? Because you're bringing it up from groundwater, it's coming up through the plants, it volatilizes and then gets deposited. And is it staying relatively local then? Because on that other figure you show where you're showing the rice paddies and arsenic concentrations, they match up quite well, which would make me believe that that's probably true, but yet there's some transport.
Yeah, I guess that's my question also. What exactly is the mass balance in this system? I guess one thing is, a lot of my previous research has mostly been on the groundwater arsenic side, like on the water resources side, and always thinking about the localized sources to this paddy environment. But I think what happens is a lot of people think it ends at the surface or the plant and don't account. Maybe it's a small fraction, but for example, maybe you do have of course I think where you're really irrigating with high arsenic in those hotspots, the most dominant will be from the arsenic in groundwater. But because the deposition and transport in the atmosphere is so heterogeneous, maybe you're saying, oh, I don't irrigate my field with high arsenic groundwater in a different location, I should be fine. There should be no sources of arsenic to this environment. But actually there is, but we can't see it. So I think there's really a spatial heterogeneity that whenever we also do the total mass balance approaches with box models, we're not really capturing that. And in the end, when you're at an individual rice field or an individual person in this location, then it really depends on all the interactions of the environmental sources at once.
That's interesting, thanks.
This will be my ignorance of chemistry, but are there isotopes, stable isotopes of arsenic?
There's only one isotope of arsenic.
You
can have radioactive arsenic.
Yeah, I was just kind of wondering if there are other ways you could tease out emission sources or fractionation or labeling. I don't know.
Yeah, I think so far what Esther did was she measured other things in the sources and tried to correlate or do principal component analysis with known sources of the other cations or things.
I wonder, are there any high frequency resolving measurements? when you think a lot about flux measurements, I'm always looking for.
Oh yeah, so that's a good question about flux. There's some flux measurements that have occurred, but nothing like what's been done for mercury. Especially, well, it's understandable because mercury is like, very like neurotoxic. But there's no like kind of flux towers for arsenic. But we did propose to collaborate with another person in Switzerland who also does these flux measurements in also rice paddy fields, but he only did one experiment like once in like 3 spots in one rice paddy field. So we proposed with him to like do it in a larger area over a longer time.
Really interesting.
Yeah, and what we suggested we could like try to pinpoint spots that would be interesting with the model. Yes.
How much of the arsenic gets into the rice grains themselves?
Yeah.
Is it detectable?
Yeah, it is detectable. There's like, plants are not my area of expertise, but there's a lot of studies out there on rice and arsenic in rice. Actually, arsenic is mobilized because there's a silicon transporter, especially specific to rice plants, that can also easily uptake arsenic, which is why it ends up in the arsenic itself. And a lot of it ends up in the grains of the rice, especially the husks on the outside of the rice grain. So this is why brown rice has more arsenic than white rice, just so you know. But I think for here in the U.S., there's all the regulations on the rice, so I wouldn't be so concerned about arsenic in rice. There's even like a lot of studies out there that go to the supermarket and buy rice and then try to measure arsenic in the rice grains. So I think we shouldn't be so concerned about it here, but it's definitely a pathway.
Anything else? Anything online? Nope. All right. Or not? Yeah.